Am I audible and can you see my slide? Just write in chat box that you are seeing my slide and I'm audible. Thank you. Okay, so let me start. So a very good evening and welcome to the course which is analytical chemistry, and this course is taken by Professor Debasis Ray, and I am Satya Prakash Yadav, PMRF scholar from August 2025 to present, and I am currently working with Professor Satish Patil, Solid State and Structure Chemistry Unit, Indian Institute of Science, Bangalore. So, a brief introduction about this course. So, we are today is the final problem solving session. So, in week one, we have solved the uh, problems based on chemical and materials analysis method. In week two, we have seen what is the role of analytical chemistry and their techniques. In week three, basically, we have discussed the problem based on chemical equilibria and what is the basis of chemical analysis of the chemical equilibria. And in week 4 and 6, we have solved the problem based on spectroscopic method, basically UV visible, IR spectroscopy, and fluorescence spectroscopy. In week 7 and 8, we have discussed the problem based on thermal methods like TGA, thermogravimetric analysis, DTA, differential thermal analysis, and DSC, differential scanning calorimetry. From week 9 to 11, we have solved the problem based on electrochemical methods. There we have seen the different kind of electrochemical technique like cyclic voltammetry, different kind of voltammetry and amperimetry, and what are the application of this technique in the uh, analytical chemistry. And today is the final week where we will see what is the application of the analytical chemistry. So, uh, in the lecture number 56 means week 12 first lecture. So, professor has discussed about what is the oxygen sensor and uh, how the modification of electrode leads to the sensing of oxygen in glucose. So, professor has discussed about the what are the key things we have to keep in mind about the oxygen sensor, and based on oxygen sensor, professor has extended it to uh, glucose sensor, and then little bit he has um, introduced about the bioelectrochemistry. And in lecture number 58, he has discussed the bioelectrochemistry in detail. So bioelectrochemistry is the study and application of biological electron transfer processes. So I will be discussing in the problem solving question based on bioelectrochemistry. And lecture number 59, he has discussed about the microbial fuel cell and microbial electrochemical cell. So these things he has discussed. So this there is no question on this microwave fuel cell and electrochemical cells. But at the end, I will briefly discuss about what is the microwave fuel cell and electrochemical cell. And in final lecture, he has basically shown you what are the different types of application of analytical science in from industry to pharma industry. Okay, so now the first question is of the assignment is what is the determined by the Clark oxygen sensor? So it is option A is dissolve O2 in blood, option B is dissolve CO2 in blood, dissolve CO2 in other body fluids, total amount of oxy compound present in the human body. So anyone can write in the chat box what is the probable answer for this question. Then I will go into detail about the Clark oxygen sensor. Can anyone just write in chat box what is the answer for this question number 1? Okay, ma'am is saying the total amount of oxy compound present in human body. Any other opinion? Okay, so let me tell you what is the what we will determine the Clark oxygen sensor. So basically, your option is A. Basically, B. Determine the dissolved O2 in blood. Now the question is how and what are the key thing about this. So first of all, what is the Clark oxygen sensor? So basically, it's a silver platinum electrode. Right. So in this, there are two electrode. What is one is the platinum disk electrode, which act as your working electrode, or it's also we say cathode. And we have one ring-shaped silver ring, which act as a counter electrode and reference electrode. And this we consider as anode. So in this, we have cathode platinum disk electrode, we have counter and reference electrode as the anode. Now, these platinum disk and ring shaped silver, uh, this counter electrode and reference electrode, which is silver wire and platinum disk, these are dipped in the KCl solution, which is act as your electrolyte. Okay, and we have one very important things, that is the replaceable membrane, which is of 10 micrometer thick and permeable to O2. Now the question is here, what is the role of this permeable membrane, which is basically made up of either polyethylene or PTFE, which is polytetrafluoroethylene, or cellophane. Okay, so these are things. Now, what is the role of this replacement membrane? So basically, this membrane is is the one which only allow oxygen to pass through it and reach to the working electrode. Right? Now we can determine by the use of technique of any polarization technique. That is the technique we have to keep in mind. Now, what are the reaction happening at the platinum and silver? So basically on silver, your silver takes Cl minus, so it will form AgCl plus electron. So what happens? Our silver will oxidize to silver plus ion, and that silver plus ion will take Cl minus from the electrolyte and form AgCl and give one electron. What happens to the cathode, which is your platinum? So on platinum, basically the oxygen will take 4 proton plus 4 electron. This will give 2 water. Now, so to balance it, we write it here 4 AgCl and 4 electron. Right? So, what is the concentration of O2? We can determine through this. So, your current, whatever we are generating, it is basically proportional to partial pressure of oxygen. So, this is all about your Clark oxygen sensor. So, basically here, what we determine by the Clark oxygen sensor? we determine the dissolved O2 in the blood. This, the further extension of this is the glucose sensor, which I will discuss in the next class. Like next question. One question. What is the correct statement for RD? It's a hydrodynamic working electrode. The electrode can be rotated during the experiment. It is used to perform steady state study of redox reaction. DR. Can anyone just write in chat box? What is the answer for this question number 2? So what is say D? Any other opinion? Okay, so the correct answer is D. Now, what is the rotating disc electrode? So, first of all, we know that CV, right? In CV, we have three electrode system. One is, suppose this is platinum disc electrode as a working electrode. We have some counter electrode and we have reference electrode in between, right? So this P2 CV. Now, how it is different from the RD? So in RD, what we have? We have same a working electrode, same like counter electrode and but working electrode has some characteristics. So what we have here? We have a platinum disc and surrounded by non-conducting plastic. The only thing is that it will rotate. Now, once it rotates, so the first thing is that the hydrodynamic working electrode, and this electrode will continuously rotate. Now in CV, we have diffusion limited current, right? Diffusion limited. Here what we have? We have one steady state, right? So a continuous flow of electrolyte from the solution will go to the working electrode. So in CV, what is the curve like? Will go like it will go to maximum IP and then it will drop it. But in RD, what happens? Because we are rotating the solution, so a continuous flux of your analyte is coming to the electrode. So what happened? It will also start from here only, but after that it will be flat. This is called steady state, right? So this is the difference between the CV and RT. Now this is called kinetic limited region, this is called mixed region, and this is called diffusion limited current. This is the diffusion limited current, right? So it performs to do the steady state study, because here we have a continuous flux of analyte which is coming to the working electrode. So this is the RD, and this is how it is different from the CV. Right. So only thing is that here working electrode is rotating. So if you want to find out what the parasitic reaction, there is called a rotating ring disc electrode. So in working electrode, we have one platinum disc. After that, we have one ring. So whatever reaction is happening on the uh, 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 working electrode, the opposite reaction will happen on the ring electrode. That's how we can manage. So it will be like this. So suppose your reaction is happening this way on the working electrode. So on counter electrode, basically it will be like this. This is called ring electrode, and this is your working electrode, correct? Like this. So this is the all the things how CV is different from the rotating distillation. Now the question 3 is that glucose plus oxygen will give hydrogen peroxide and gluconic acid. What enzyme is used in the above reaction? Maltase, glucose oxidase, trypsin, or catalase? Anyone? What is the answer for this question? So this question is basically from the glucose sensor. Then I will tell what is the different part of the glucose sensor and what is it. Can anyone just tell what is the answer for this question number 3? C. Trypsin. Okay. Any other answer? Okay. So the correct answer is B. So what trypsin do? Trypsin basically is a this is an enzyme which breaks the protein to the amino acid. Alright. What catalase do? Basically, it decompose the H2O2 to the water. What maltase do? Basically, it catalyze the hydrolysis of maltose to glucose. And what glucose oxidase do? Basically, this is the first thing is that the very important. This is the oxido reductase, means oxidation and reduction will happen. So what happens? Your glucose will oxidize to the gluconic acid, and your water oxygen will uh, oxidize to the H2O2. So this will catalyze glucose to gluconic acid, and oxygen will go to the H2O2. So by the name only, this glucose oxidase means it will oxidize glucose to oxidase. Maltase means somehow it related to the maltose. So these are the trypsin, basically protein to amino acid. Catalase means it will decompose the water into hydrogen peroxide into water. Right. So this is the uh, glucose sensor picture. So I will come in the next lecture, next question in the detail. Okay, where it will come all the detail. So I will come. Okay, what are the membrane of the electrochemical glucose? Why are the membrane of glucose sensor more complex? So first, outer membrane are permeable to glucose, impermeable to protein and other constitutive fluid, consist of 3 layers and prevent enzyme from leaching out. All of the above. Can anyone just answer? Okay, Sunil Pawar is saying D. All of the above. Any other answer? Okay, so Sunil Pawar is correct. Why this is complex? Because there are three layers. One is outer layer, inner layer, and the catalyst layer. So first thing is that you have to make three layer. That's that complex. And second thing is that membrane should be permeable to uh, some some compound and impermeable to others, like that. So your outer membrane, through outer membrane only glucose can enter, but other like protein and the other constituent of blood should not. That is the first layer. Three layer, it's in three layer is there. This is called catalyst layer, where your glucose, so here oxygen will also come. So beta glucose and oxygen will come through the outer layer, where glucose will go to the gluconic acid and oxygen will go to the uh, hydrogen peroxide. So first thing is that your glucose sensor is basically a modified Clark electrode, right? And this is also called enzyme electrode. So in previous uh, as a problem solving session, the one question is also there, like what is the enzyme electrode, what enzyme should be, like that one question was there. So this is a enzyme electrode and it's a modified Clark Clark oxygen sensor. So in Clark oxygen sensor, what is that? Basically we had this two electrode and we have a permeable membrane which only allows O2 to pass. Now here what we had the, here also we have two electrode. One is the working electrode, another is the counter electrode and reference electrode. But working electrode is slightly modified. So here there was only one membrane which only permeable to O2. But here there are three layers. In first layer we have only this your glucose and oxygen can enter. In the second layer, what happens? Your beta, whatever D glucose is there, plus oxygen is there. So you have this uh, glucose oxidase, glucose oxidase. So this beta glucose will go to the gluconic acid, and your oxygen will go to the hydrogen peroxide, right? So if I write, so this is the thing. Now what happens? This the in inner layer only H2O2 can pass. That is the another important things. Here only H2O2 can pass through this membrane. So in inner layer only H2O2 will be there. Now what will happen? Your H2O2 will go to the 2H plus O2 plus 2. Now we can find out what is the H2O2 concentration by the electrochemical technique. Now the whatever current correspond to the H2O2 is basically the concentration, like current whatever I is proportional to the C H2O2, and this is will be proportional to C concentration of O2, which is concentration to the glucose. So this way basically the current will be proportional to the your glucose. So by back calculation we can find out what is the concentration of glucose. And one more thing is that, so here if we write another way, so you can write it H2O2 plus 2OH minus, it will be 2 water molecule plus O2 plus 2. This reaction can be also written. So we know, and on counter electrode side what will happen? Your silver will go to this silver plus. So basically silver will form silver chloride, and silver will take Cl minus and it will form silver chloride plus electron, like that. And this reaction correspond to zero point 0.6 volt versus AgS, right? So this is all about the glucose sensor, where the basically here two electrode. The working electrode has three layer. In outer layer only beta glucose can go, and in the middle layer, which is called catalyst layer, where glucose will go to the gluconic acid and oxygen will go to the hydrogen peroxide. And from the middle layer to inner layer only hydrogen peroxide can go, which is reached to the electrode. There hydrogen peroxide will uh, reduced to the water, and that whatever oxygen will be remain, it will again go back to the middle layer. So that hydrogen peroxide concentration will be find out by the current, and that will be proportional to your oxygen concentration and glucose. So this is all about the glucose sensor. Uh, like this is all about the glucose sensor. So these things we have to keep. Okay, this is done. Oh. Now, question number 5. Which of the following option is the correct for the phosphorylation? Phosphorylation. So, ATP converted into ADP plus pi, ADP converted into ATP, glucose is produced, AMP is produced. Can anyone just write the answer in the chat box? Anyone? What is the answer for question number 5? Sunil is saying ATP converted into ADP plus PR. 5. Okay. Any other opinion regarding this question? Yeah, I'm saying B. So yeah, and the correct answer is B. Now let's see what are the processes happening. So first thing is that what is ADP and what is ATP. So ATP and ADP are differ by one phosphoric acid group. So phosphate group, basically one phosphate group. So ADP is this unit total is called adenosine. So adenosine diphosphate. This is adenosine triphosphate. Now what will happen? This your ADP to ATP formation. This requires energy, right? That basically we take energy by the your photosynthesis or any biological process. Through this ADP will convert to the ATP. Now this is more energy state. So what will happen when the hydrolysis of ATP will happen? So what will happen? One phosphoric acid will give release, and this will give the your energy to work your body to work. So whatever energy we are the energy is coming basically from the ATP to ADP curves. So this is called hydrolysis of phosphate group. So what is the phosphorylation? So phosphorylation is basically your ADP. So attachment of one phosphoric acid to the ATP. So one phosphate group will attach to the ATP. This is called phosphorylation. It's like the word hydrolysis, hydroxylation, hydrolysis, like that. If something kind of like dehydrogenation, term is written, then you can say that the removal of that dehydrogenation is removal of hydrogen like that. So this answer is not correct. This is the correct where we basically ADP to ATP conversion basically be attached one phosphate group. So this is called phosphorylation. Correct? So I think Sunil, and now you got it. What is the phosphorylation process? It's like just a phosphate. So ADP will do by the phosphorylation, it will convert to the ATP. So one phosphate group is atom. So this is adenosine diphosphate. This is adenosine trans. So this is the high energy bond. So this is very high energy bond. Once we hydrolysis it, so basically we get the energy. Right. So this is the like one picture to just and for the understanding purpose. Okay. Now the question number 6. Outer layer of the electrochemical sensor is thin film of KCl, cellulose acetate membrane, polycarbonate film, or glucose oxidase enzyme. Can anyone just tell like what is this answer? What is question number 6? The outer layer of the electrochemical glucose sensor. So if you have seen the lecture, professor's lecture of glucose sensor, so there he has discussed about what is the outer layer of electrochemical glucose and what is the importance of this outer layer. Anyone can just write in the chat box. What is the answer? B. Cellulose acetate membrane. Okay. Thin film of cells. Any other answer? Oh, ma'am is saying the glucose oxidase enzyme. Okay, so the correct answer is the polycarbonate film. So I will just go back to that slide. So this is your outer membrane. So which is made up of the polycarbonate film. So that property of that polycarbonate film is it will only allow to the beta glucose. And other options are glucose oxidase enzyme. So glucose oxidase enzyme, this is in the middle layer. The work of this glucose oxidase enzyme is basically it will convert beta D glucose to gluconic acid and oxygen to H2O2. And that thin film of other option is what? Thin film of KCl solution. So that thin film of KCl solution basically will be the inner layer where your H2O2 is converting to the uh, oxygen, right? And it is giving the where the electron transfer is basically happening. So they are only your KCl solution. And other this cellulose acetate membrane. So I am not aware about that. What is the property of cellulose acetate membrane? Whether it will be only permeable to the glucose or not. Okay. So the correct answer is polycarbonate film, which has property to like allow only uh, glucose but impermeable to the protein and other constituent of the blood. Okay, so I think now it will be clear what is the outer layer of electrochemical glucose. Okay, so now I will go to the question number 7. Okay, Little bit more about this polycarbonate field. Basically, it's a porous membrane that will allow to pass the bigger molecule glucose, but prevent the another smaller fluid constituent to pass through this layer. That. Okay. Now the question number 7. Fill in the blank with the suitable option given below. H2O2 plus 2OH minus O2 plus 2H2O plus 2 left. The above reaction takes place in the middle layer of the electrochemical glucose sensor, cellulose state membrane, inner layer or electrode surface. Can anyone tell the answer for this question number 7? Where it happens? That this reaction? Anyone can write in the chat box what is the answer for this H2O2 reduction to the uh, H2O2 oxygen to oxidation to O2. E middle layer. A is middle layer. Okay. Any other answer? Okay. Yeah. Sunil Pawar is correct. I think I should go once again. So what are so first is that the outer layer. Outer layer is just a polycarbonate carbonate film. Okay. So this will allow your glucose and oxygen. That is the first layer. In the middle layer, we have your enzyme which is called glucose oxidase. What glucose oxidase will do? This glucose oxidase will convert your glucose to the gluconic acid and oxygen to H2O2. Now you have third inner layer. So through the inner layer only H2O2 will pass. That is the inner layer. So that H2O2, now the H2O2 will be converted to the oxygen plus water plus 2 electron transfer. Right? So it will take 2 OH minus. So this is happening at the electrode surface because electron transfer only happen at the electrode surface. So this is the correct answer. So ma'am, the middle in the middle layer, what is happening? Your glucose oxidase is basically converting glucose to gluconic acid and oxygen to the H2O2. So this is in the middle layer. So the question is that where this reaction is happening, H2O2 is converting to O2. That will happen at the electrode surface in the inner layer. Okay, so here the question is that the above reaction takes place at, so that it will takes place at the electrode surface. Okay, yeah. So now I will go to the question number 8. So what types of bacteria desulfovibrio gigas each? So this is again from the lecture where the professor is discussing about the bioelectrochemistry. Can anyone just guess? glucose oxidizing bacteria, carbonate reducing, nitrate reducing, sulfate reducing. Okay. Saying B, sulfate oxidizing bacteria. Any other answer? Okay. So the answer is this is a kind of sulfate reducing bacteria. So your D-sulfobaric gigas, which is also called D gigas, people used to write D gigas, is a sulfate reducing bacteria. So basically, uh, uh, it is a sulfate reducing bacteria, which is kind of like bacteria that can perform in a aerobic respiration. Okay, so anaerobic means in absence of oxygen, right? So here what happens? Your SO2 minus will basically reduce to the H2S. That is by this D gigas bacteria, right? So okay, so this is the correct answer. That it is a sulfate reducing bacteria. It's not sulfate oxidizing bacteria. Okay, this is again like all the uh, biology question. So what bacteria do? Okay, so the mechanism is like complex. So I'm not going to the detail how it is reducing into sulfate to the H2S. Now the question number 9, which is fill in the blank with the suitable option given below. So this is called orthodiphenol. Diphenol plus half O2 is going giving orthoquinone plus water. The above reaction takes place in presence of which reaction? Any answer? Okay, saying B. Catechol. Okay. Yeah. Any other opinion? Yeah. So the correct answer is catechol. So it's also like for us. So this is what is the monooxygenase. So this is the a copper based enzyme. So what is happening? First we have this ortho orthodiphenol. Okay. So okay. So first we have phenol, right? So this is a like basis for the kind of phenol sensor. Now professor has already discussed like how the phenol how much like phenol sensor is important. So the phenol, first we have here the catalyst, like enzyme, which is called monooxygenase, right? So insertion of one oxygen, so it will form your ortho diphenol. Now the other enzyme, which is the answer, this catalyst, this will give you the quinol. Okay, so here half O2 and then here half O2. So this will give, and this one of the important property of this has a strong optical absorbance, right? So the concentration you can also like find out through the any UV visual technique because it has a strong absorption property. Now what is kinase? Kinase is the like catalyst which is like which catalyze the transfer of phosphate group from high energy phosphate donating molecule to the specific substrate. And the catalyst we have already told you, like it's a like decomposition of hydrogen peroxide to the water and oxygen. Okay, so yeah, I think Sunil is right. The enzyme which is converting orthodiphenol to orthoquinol is the answer is B, catechol. Okay, now I will go to the like last question, which is question number 10. Fill in the blank with the suitable option given. Phenolic biosensors are used in wastewater treatment process, food like milk product, medicine, and biotechnology. All of these. Can anyone just write in chat box? Okay. Okay, so this is like phenolic biosensor are used in wastewater treatment process, food like milk product, medicine, and biotechnology. So the answer is D, and the professor has already told you all about the phenolic biosensor. So in wastewater treatment, basically it's used to detect the contaminate in the surface and groundwater. In medicinal and biotechnology, this is used to the catecholamine, which is in blood, brain, and cell, and also it is select like used to detection of cell membrane and sensitive level detector in the enzyme immunology. And in food, like basically the red wine and milk to control the food quality. So all are the application of this phenolic biosensor. Now I will just go a little bit in detail about the this oxygen sensor. I have already told you how what is happening, so you can just go and see this because I have already told what are all the like enzyme based sensor, what is the reaction happening, bioelectrochemistry. So one that thing is that that question didn't come on this topic. It's called microbial fuel cell and microbial electrolysis cell. So first thing is that what is the microbial fuel cell? So in this, what happens? We are producing the electricity, and in the microbial electrolysis cell, we are giving the electricity to like to the conversion of the chemical from one state to other state. Okay, so this I will just show you like uh, so what are the main thing. So in MFC or MEC, which is called microbial fuel cell and microbial electrolysis cell, in this typically which is called bacteria, they break down the organic molecule which is found in the wastewater or from any like contaminated under a anaerobic condition. So here there is no oxygen. And during this, there are three things remain. Three things evolve. One is the electron, another is proton, and carbon dioxide. So carbon dioxide we can collect it and used for the other purpose. Electron can travel from anode to the cathode. I will show in the next slide. And to the cathode, and proton travel through the membrane to the cathode. So I will show you what is happening in the microbial fuel cell electrolysis. So first thing is that wastewater we are supplying to the anode. So what is happening here? Wastewater is basically generating electron, CO2 and proton. So what is happening? Your electron is traveled through the external circuit, then it will lighten the bulb and it will come to the cathode. So here what is happening? Your oxygen is coming and basically it's like here oxygen reduction catalyst is there and it will reduce the oxygen. Now for that we need proton and this proton will travel through the membrane and it will come to the cathode. So basically O2 plus 4H plus 4 electron, it will give O2 plus 4H plus 4 electron will give 2H2, right? So two water molecules, so this can be so whatever that excess oxygen is there, this can be exhaust and CO2 can be calculated. What is the effluent is basically the waste liquid, so we can collect it here. So this is all about microbial fuels. So here what is happening, we have wastewater we are giving and that bacteria. So if we see here, so this is your electrode, this is your called your electrochemical active microorganism bacteria where reaction happen. So organic material will be come here and it will convert CO2 H2 and that electron will travel through the external circuit. So this is kind of, here we have the chemical reaction to the electricity. It's a kind of battery under discharge condition. This is called fuel cell. Now in electrolysis, what is happening? We are supplying the power to happen the reaction. So here what is happening? So wastewater we are supplying. Upon the so like in this case reverse is happening. So wastewater we are due to the electricity, wastewater will producing electron, proton and CO2. CO2 can be captured, proton can travel through the membrane to cathode and electron through the external circuit. The important thing is that this is used for the hydrogen production, methane production. So these are the application like you can go through in the literature. So what is happening now? Whatever that H plus is coming, so H plus, 2H plus will go to the H2 like 2H plus 2 electron will give you H2 and this hydrogen you can collect it. So this is basically a hydrogen production system. So in this we are giving the electricity and producing hydrogen and so there are other processes also. Application, this one of the important process is copper recovery. So you can go ahead and see that and these are the phenolic biosensor. So where like what are the like possible applications? So why the phenolic biosensor is very important. So that last question is there, like food product, especially like olive oil, wine, herb, spicy. So these are the possible application. And that phenol oxidation by water producing oxidase and oxygen. And the one of the most that enzyme which is tyrosinase. It's a copper containing monophenol monooxide. So that is mainly responsible for your uh, uh, like reaction. So that is the first thing, where the phenol is going to the orthodiphenyl via monooxidase and then orthodiphenyl via cataclase will go to orthoquinone and right. So here that one thing is that this is wrong. So basic, sorry, this is wrong here. So this should be like this. Wait. Okay. So these are the application: environmental control. So wastewater treatment, medicinal. We have to find out this kind of uh, chlomine in blood brain. This is the in the last question, and food quality, like we have other like pointed in milk product. So this is the possible application of phenolic biosensor, and these are the common like what is called application for this. uh, sorry. Common application like professor has discussed in the detail in the lecture number 6. So you can go ahead and read what are the powers. I will upload this PPT in this video lecture as well. Okay, so let me stop the record.